So Salim, Simone, Ikram and Mohammed, Nahed, sorry, could you please come to the stage, take a seat here? Thank you very much. Thank you everybody for taking your seats. The purpose of this session is to highlight some of the research projects happening at the Center for Desert Agriculture that have impacts on food security in Saudi Arabia. Each speaker will have 10 minutes to present. I will use the same 1-minute warning mechanism as for the last session, and then we will have a panel discussion at the end. To begin this session, we have Professor Salim Al-Babili. He is Professor of Plant Science at KAUST. Dr. Babili's research aims to generate crops with improved agricultural performance and enhanced nutritional value. He will present on the Striga project in Africa and its relevance to Saudi Arabia. Thank you, Dr. Salim.
Thank you very much for the nice introduction. Today I'm going to speak about our project in which we translated basic plant biochemistry into agricultural application. What you see here is, okay. What you see here is a sorghum field in Africa. Besides sorghum, you see beautiful flowers that are actually of a plant that's called Striga hermonthica, or witchweed. That is a quite dangerous plant because it affects the yield of cereals in many parts of the world, particularly in Africa. It is a parasitic plant. Indeed, this plant is one of the 7 major threats to global food security. It's causing annual losses ranging from $7 to $10 billion every year. As I said at the beginning, it's particularly affecting agriculture in Africa. You see the distribution of this parasitic plant. It's present in sub-Saharan Africa, in Eastern Africa, and in Southern Africa. Striga is a so-called root parasitic plant. This means it relies on a connection to its host, to the roots of its host. Usually seeds of this parasite remain viable in the soil for a long time, waiting for a suitable host. They recognize the host by chemicals such as strigolactones. You see the structure of these chemicals. These chemicals are usually released by the host plant to establish communication with beneficial microorganisms. Once they perceive this signal, they germinate, they build a bridge to the host that is called haustoium. Through this bridge, they can absorb water and nutrients from the host. The result is, of course, devastating the yields and actually inhibiting the host growth. Most part of the damage happens before you see the plant above ground, when the seeds are germinating and building the connection to the host. One Striga plant can produce up to 200,000 tiny seeds that remain viable for 15, 20 years. And this actually makes the combating Striga a very difficult issue. If you want to combat Striga, you need first to get rid of these seeds that have accumulated over the years in infested soils. One possibility to do that is the so-called suicidal germination. You apply, in the absence of a host, chemicals, strigolactone analogs that can induce seed germination. Striga is an obligate root parasitic plant. This means it cannot survive if there is no host in the vicinity. Therefore, seeds would germinate and would die. We call this suicidal germination. That is, in principle, this has been a theoretical option. No one really tested it in reality. I'm plant molecular biologist, biochemist. I was working on the biosynthesis of strigolactones. Thereby, we identified a simple strigolactone that we called Carlactone. You see the structure here. Then we thought, why we don't use the structure of Carlactone to develop a new strigolactone analogs that can be used for the suicidal germination. I was lucky to present this idea to Mr. Gates when he visited KAUST. After a while, we got a proposal approved to test our hypothesis and strategy in Africa. We started this work a few years ago in Burkina Faso. This work is, of course, was also supported by KAUST. To apply the chemicals, we established a rain-fed protocol for rain-fed agriculture. We applied the compound after a first rain. This first rain would also induce the so-called preconditioning in the seeds. Second rain would dilute the compound in the soil and bring it to the seeds, where it induces the germination and causes the death of emerging seedlings. We got very promising results. And we went to a second phase of this project in which we are continuing improving this suicidal germination technology. In addition, we are applying growth promoting compounds that help the plants to escape Striga infestation. One of them is zaxinone. You see here the effect of varying zaxinone concentration in rice. And we are also working on establishing tools to improve permillet as a crop to produce resistant varieties of this plant. Permillet is a very important crop for arid and semi-arid zones. Actually, it has become an orphan cereal in Saudi Arabia, although it is a classical one. I'm trying to lobby for this plant. We have, in the meanwhile, probably the first transgenic plant in which we are doing functional genomics of permillet. And as I said, we are also establishing the genome of this cereal. Of course, this work has been done, is still being done by a consortium with international collaborators, in addition to my collaborators within KAUST that are making significant contributions to the progress of this project. And at the end, I want just to leave you with this short movie about our project. Thank you very much.
Professor Salim, you get a gold star for being early finishing your talk. A gold star.
You told me 10 minutes, 10 minutes, 10 minutes.
Okay, thank you, Professor Salim. Our next speaker is Professor Simone Krattinger. Professor Krattinger is Assistant Professor Plant Science at KAUST. Professor Krattinger's research consists in the translation of basic knowledge into plant breeding with the ultimate aim of developing highly resistant crop cultivars. So will you please come to the stage and present on your research? Thank you.
Okay, thank you, Paul, for the introduction. I also tried to get the gold star. So today I'm going to talk about, not my research, this is one of the three basically translational research projects that Rod Ring mentioned this morning that we're doing in the center. I think it's a quite bold project, but it will be very impactful. And at the end of this project, in the medium to long term, what we want to do is we want to generate new crop plants that are suitable for the desert environment of Saudi Arabia. Now, let me place the key message right at the beginning. We need more plants, or in other words, we need more crops, or we need new crops. Now, this is not my statement. This is something that scientists and breeders have realized for a long time. Our global food production, and hence also food security, really depends on only a handful of plant species. Now, if you look at this in more detail, there is about 30,000 edible plant species out there, but only a very small fraction of this is used in modern agriculture today. So we have left behind a huge diversity, genetic diversity, that we don't use in agriculture. So if you look at the top of this pyramid, you see that only 3 species, these are rice, wheat, and maize, account for 50% of the calories that we consume. So in other words, global food security or the global food system really depends on our success to grow these 3 crops. Okay, we have talked a lot about this, the challenges and risks associated with this. So we have to realize that, again, when we talk about food security, we really also have to talk in one way or the other about grain production. Grain production, so the cereals, they account for most of the calories that we consume, and they use most of the energy that we use in agriculture. So here again, you see the major cereals: rice, wheat, maize, barley, millets. Again, rice, wheat, and maize, these are the dominant cereal species or crop species. The problem with them is that they're not really suitable for a harsh environment or for an extreme environment like this, the environment in Saudi Arabia, because many of these species originate from rather wet and humid regions. For example, rice is from the subtropicals was domesticated in South Asia, maize in Central America. So again, they are not really suitable for a desert environment. Now, there's 2 ways how you can deal with it. One way is what breeders try to do and have done quite successfully, which is to try to breed for more drought-tolerant and heat-tolerant rice, wheat, and maize. Our approach is a bit different, and I think it's bold. So what we want to do, we want to go out again into this untapped diversity of the 30,000 potentially edible species and repeat the process of domestication. So take a wild plant that is highly adapted to a desert environment and turn it into a high-performing crop. So if you look at the top, domestication is a process during which our ancestors converted wild plant species into modern crops. This is a process that started 10,000 years ago, and that really changed the way these plants look like. Okay, if you look at wild maize, which is teosinte there, it has a very small cob, and it produces only about 5 or 10 very hard and unedible seeds. And over time, our ancestors have changed this wild maize into what we know today is maize, produces these large cobs and hundreds of seeds that are tasty. And they have done this by selecting for spontaneous mutations and versions of plants that have been selected for. Now, this was a gradual and slow process that took hundreds or even thousands of years, okay? And again, as a result of this, only a few plant species were domesticated. So again, we have left behind this untapped diversity. And it's quite an interesting thought. As modern as agriculture is today, it's still based on decisions that our ancestors took 10,000 years ago. Why have they domesticated wild maize, and why did they not domesticate other species? So what we can do today is we go out, we look for wild crop relatives that are adapted to this harsh environment. They will have unfavorable traits, as most wild species have. They might have seed shattering, they might not be highly productive. But today we can turn this into highly performing crops within 10 to 15 years with the technologies we have in hand, such as genomics, tissue culture, and genome editing in particular. So this is the vision we have, that within 10 to 15 years, we can come up with new crop species that are suitable for Saudi Arabia. Now, I just want to show you a few examples. So here, for example, this is a field of fonio millet. Fonio millet is an interesting crop. I call it a semi-domesticated crop or neglected crop, similar to pearl millet that Salim mentioned before. It's a crop that is cultivated in West Africa, that it's consumed in West Africa, but that still looks very much like a wild plant. So we have done a trial. This is a trial at the KAU experimental field station in Had al-Sham, which is about an hour's drive from here. And you see that this crop already does really well under these harsh and hot conditions. It uses about a third of the water that is used to grow wheat and maize, for example. It's known as the fastest maturing cereal on the planet. It can produce seeds or grains in under 2 months under optimal conditions. So here you see the grains of fonio millet. They're quite small compared to rice, maize, and wheat. And this is such a plant. This is an ideal example, a textbook example of a plant that thrives in harsh environments. It grows in the Sahel zone in really hot and dry conditions, and that we can turn into a high-performing crop that can do well in the desert. We're working on other species as well in the initial phase. For example, we're working on an Asian wild rice or rice octata that Rod Wing is leading this, that can grow in salt water and that is also used by local communities for food. Mark Testa and Vanessa Melino are working on Saliconia, which is an extreme halophytic plant that can really thrive in seawater. Okay, all these plants have negative or are not really suitable for modern agriculture. They need a lot of adaptation to turn them into high-performing crops. But these are our initial targets. And then, of course, we also go out and look for new crop species, new wild crop relatives that we could turn into highly performing crops. Now, these are the work packages. Just to give you an overview, what are we doing in this initial phase? We are essentially establishing all the resources that are necessary to establish, I call it a molecular breeding program for these plants. These include generating high-quality reference genomes, sampling the diversity across the species. Very important, establish gene editing methods, characterize those plants in detail, and also a very important work package is this work package 6, which looks at functional synthetic microbial communities. We know that these microbial communities are very important to help the plant growing. Okay, now what's the potential impact and what are policy considerations that we might need for such a medium to long-term project? I think, again, it can be and will be quite impactful because what we are doing at the end of the day, we're establishing new crops and we can establish entire new cropping systems for sustainable, maybe even saltwater-based agriculture in Saudi Arabia. And we can significantly reduce the freshwater footprint of agriculture in the kingdom, okay? And with this, I mean, we talked a lot about this, right? Drop for crop, we can increase local food production. And what I think here is the first point, unlock marginal land for crop production. And by this also increase biodiversity in agriculture. At the beginning, I showed you we need new plants. And I think KAUST, the kingdom, can lead this change that is needed. What we need for being successful really, and where the kingdom might help, is actually to really have a good legal framework to exchange plant material. How can we get new wild crop relatives into the kingdom? What we want to do, so also a vision is establish a global halophyte seed bank, so a big germplasm collection with new potential crops for the kingdom. We need a good legal framework for research on genome-edited plants. We need experimental farms or agricultural stations where we can do our proof of concept work. Ideally, again, this would allow genome field trials with genome-edited plants. And I think what we have, and this is, I'm really fortunate that we have funding streams at KAUST and in the kingdom that allow us to perform this long-term vision. Again, it will take 10 years, maybe 15 years until we succeed. But I think really, again, at the end of the day, this can be really, really impactful. And with this, I hope I could give you a consideration, an overview of this project. I didn't hear the bell, so I assume I get a gold star, Paul. So thank you for your attention.
2 speakers, 2 gold stars. Dr. Nahed. Our next speaker is Dr. Nahed Mohammed. Dr. Mohammed is a postdoctoral researcher in KAUST. Her research focuses on studying plant genomes with the ambition to contribute her research to solving the 10 billion people question, which is a big question. Dr. Mohammed will explain how the Native Genome Project will contribute towards sustainable food security in Saudi Arabia and how these results could be accelerated with the right policies. Thank you.
Good morning, everyone, and thanks for joining us in this workshop. And thanks for the organizers to give us the opportunity to present some of the research that we do at the CDA. And it's a pleasure to introduce the KSA Native Genome Project to the audience. Today, I'm going to talk about the Saudi government efforts to improve its agricultural sector as part of the Vision 2030. And I'm going to introduce the KSA Native Genome Project with its long-term goals and the pilot project that started a few months ago. I'm going to give a brief introduction to the list of the targeted plant species and the brief plan of work. And then I'm going to explain about how the Native Genome Project contribute toward the sustainable food security in Saudi Arabia, and how we can use these results to reach the targets of the Saudi Green Initiative. I'm going to end with a summary and future direction of this project. So environment protection via the conservation of biological diversity is the central theme of the Kingdom of Saudi Arabia's Vision 2030. And Saudi Arabia has taken important steps towards the consolidating effective and flexible national food systems. And it's based on strategy on building like sustainable agricultural sector that would contribute to achieve water and food security alongside with ecological balance in Saudi. And we all know about these programs that are called Saudi Green Initiative and Middle East Green Initiative, which are huge steps for the kingdom to scale up their climate action and environment protection. So the Native Genome Project is a collaboration between the CDA, Center for Desert Agriculture at KAUST, and the National Center for Vegetation Cover Development and Combating Desertification in MIWA. So it basically aims to study many common and endangered plant species that are significantly important for their use. Some of them have cultural, religious, medicinal, and pastoral significance. So this study contributes to achieving the Kingdom vision in the field of agricultural development, and also food security and protecting the environment by preserving biological diversity, and also promoting the principle of sustainable agriculture in the kingdom and combating desertification. So the long-term goals of the project is to biologically characterize different ecosystems across Saudi that are relevant to the mission of the NCVC Center to generate high-quality genomic resources for all the native plant, animal, and associated microbial species in Saudi Arabia, and also to provide essential data as to how and why particular ecosystem operate in particular environment, such as the extreme conditions that we have here in Saudi. And that would produce a new cohort of Saudi scientists trained in the study of ecosystem biology and application to environmental protection and conservation. So the pilot project started July 2021, and the aim of this pilot project is to study a set of 15 terrestrial ecosystems categorized as threatened species, rangeland species, and common species. And the aim is to provide high-quality reference genomes for each of these species and to investigate their ecosystem with respect to metabolomes, road development, architecture, and microbiomes. That would establish a robust infrastructure to characterize hundreds or even thousands of different native ecosystems across Saudi, both terrestrial and aquatic. So the list of the targeted plant species that we are studying at the moment, as I mentioned, they are categorized as threatened species, common species, and rangeland species. Each category have 5 species. Some of these have medicinal use, others have religious value, and others are used for animal pasturing. So the brief plan of work is to do the vouchering of these plants, arrange field trips in different locations in Saudi to voucher live specimens. And then we do the plant genome sequencing. And with the help of other labs here at the CDA, we do the microbial analysis as well. The metabolomics analysis is done in Al-Babili lab, and the root development and agriculture is in Blilou lab. After that, we're gonna release and deposit all the data in NCBI for Gene Bank for public use, and also in National Gene Bank. So the vouchering, as I said, we arrange field trips with MIWA to identify a set of live voucher specimens. We tag the plants, we collect the samples, and at the same time, we preserve these samples as herbarium specimens. And the aim is to deposit these samples at key herbariums around the world, starting with the seed center here in Riyadh, in Saudi. All these vouchers will be phenotyped and RFIDs will be placed. The second part is the plant genome sequencing, which is done here in the Wing Lab. We extract high molecular weight DNA of the samples to construct libraries for long-read sequencing, and also use the RNA data for genome annotation. And everything is performed following the standard protocols established in the Wing Lab. So for the sampling expeditions, we managed to collect 44 native species from different regions across Saudi, 15 targeted and additional 29 species. These include common and endangered plant species that have cultural, religious, medicinal, and pastoral significance. These plants have been collected from different ecozones. Some are collected from mountains and highlands. Some collected from wadis and floodplains, and some from desert and sand dunes. So from Hail, we managed to collect 9 species. From Taif, we collected 4 species. From Alith, we managed to collect 6 species. And from Abha, 10 species. And finally, 15 species from Jazan. So the plant genome sequencing, I'm just going to show an example of one of the plants that we worked on, is the Juniperus procera, known as arar in Arabic. So we isolated the high molecular weight DNA. We did the QC analysis just to make sure that it's okay quality and quantity-wise for the sequencing. And we submitted the samples for the core labs here at KAUST using one of the latest technologies in whole genome sequencing. So I'm going to talk a bit about the juniper as an example. Juniper is a long-lived evergreen tree. It's a predominant species in forest and at altitudes of 1,400 meters and above. It's found in the mountains of the southwestern region of Saudi Arabia, and it's suitable for afforestation of streets and public gardens in those areas. Now, unfortunately, the juniper trees are exhibiting widespread decline with extensive tree dieback. The causes haven't been clarified yet, but many researchers have expected natural disturbances such as drought and climate change and disease and so on. And the magnitude of the dieback of juniper trees varies from place to place and exists in both lower and higher areas. So we have sequenced the genome already, and then we're gonna use the information of the population genetics to study different juniper population in Saudi to help us address the problem of the dieback and inform us on reforestation of Saudi Arabia, which is aligning with the goals of the Saudi Green Initiative. Some of the plants that we are working on are suitable for forestation programs in Saudi, such as the ficus, the tamarind, the ziziphus, and moringa. And some plants are also can be used in the desert restoration, such as these range land species. And also, similar studies could be applied on food crops, such as the ajwa date palm and hasawi rice. which also contribute to food security. So just to give a summary, the Native Genome Project goals aligns with these of the Saudi Green Initiative. This project is very important for the youth of Saudi Arabia to learn about the native species and to embrace sustainable agriculture and environment protection in their country. For the future direction, experimental station will be established in the KAUST area and in different areas across the country. And also, this list of species can be extended to include other native and endangered species, not only for plants, but also for animals and aquatic species such as the falcon, Arabian oryx, and Nagel. With that, I would like to thank everyone who contributed to this project from the CDA and from the NCVC Center. And thank you very much for your listening.
Well done. Thank you very much. Squeezed in for a gold star. Just squeezed in. Our next speaker, final speaker for this session is Professor Ikram Blilou. Professor Blilou is the Chair of Plant Science at KAUST. That plant science program aims to develop an integrated understanding of how plants can grow under extreme environmental conditions, which is obviously the sort of conditions that you might find in Saudi Arabia. Professor Ikram will describe the Fast Fit Palms program and its implications for the Saudi date industry and what actions and policies could accelerate this.
Okay, thanks for the invitation. And before starting, I would like to make a kind of observation. So it's a great summit, and it's very important that we are looking for sustainable solution for food security. But we should not forget also that we also have to go to the basics, and the basics, which would be its science. So how to combine science and how to go into maintaining and toward the long-term goals to achieve this sustainable food security. So what we have here is, you can see it like a pie, because it's almost lunchtime, and I'm here between you and lunch, and you can see this pie, which is a root, and a root is one of the most important organs for plant to grow. I don't know. I'm trying to take over the stage. So one important thing that I would like to stress, that basic science is important. Understanding the mechanism of how things work are important if we want to achieve long-term goals for food security. And a great example is the Food Institute this morning. So you have heard the great talk that was presented by David Walsh. And David Walsh was a stem cell specialist. He was a basic researcher by science researchers, and thanks to this knowledge, he translated this knowledge into a very beautiful translational application. And that is a really great example that we should keep in mind if we want to achieve these our aspirations. So what we do at KAUST, so we have a very broad programs, and these broad programs are mainly converting basic science into translational research. And we do we study many aspects ranging from cell biology, developmental biology, genome editing, and with the aim of creating tools into improving agriculture in the kingdom. And not only in the kingdom, but also globally, because it's a general question. So we need to understand biological processes if we to be able to translate them into translational solutions. So that's what we do in the lab. But for this session, I would like to focus on the date palm program that we have here established at KAUST. And what you can see here, just by understanding some very basic processes of date palm growth, we could really understand how these plants are able to cope with the environmental conditions. And these environmental conditions, is drought, salinity, and lack of water. So it's what I think is that palm is a very promising contributor to food security. They are very nutritious, they are very relevant to the kingdom, and most importantly, they are really available. So there is not a shortage of dates. There is a large variety, especially in the kingdom. So I think it's the first producer from the Middle East countries. It has a great resources. If you go to Al-Hasa regions, there is it's in the Guinness encyclopedia, and they have at least 30 million palms are in the kingdom. 20,000 hectares are cultivated with palms, and they produce at least 5 tons per day. There are at least 5,000 variety of date palm, and in the kingdom there are more than 400, and not all of them are producing fruits. We are only focusing on a few that are known, or few cultivars that are known by, or maybe were adapted to their taste. And that's why we are not really fully exploiting the potential of date palm fruit. So within the Fast Feed Palm project, what we are doing is trying to establish the foundation. So this project is not for today, it's not for tomorrow, but it's for the next generation of scientists, the next generation of Saudi, and the next generation of whoever want to carry the torch and then build the date palm and keep the date palm culture. So we have established objective, and the objectives will be to make a stable transformation, to improve the regeneration protocol for dates, to accelerate the growth, because we know that the date palm, they take at least 15 years to flower. So what we want to do is to find a way to make them flower within 5 years, and to understand the genetic diversity. Why do we have Ajwa? Why do we have Khalas? Why do we have Deglek Noor? Why does Ajwa grows nicely in Medina and not in Oman? Why do we have dates that are growing in the seashore and are still able to produce fruit? How can we exploit this genetic diversity and this variation to translate it into a more sustainable knowledge? So as a project, we use genomics, transcriptomic, phenomics, growth regulators, and microbiomes. And this is a project that is in close collaboration with members of the CDA. So all the members of CDAs are involved here, and each member is taking care of one aspect of the project. And what would be the outcome? The outcome would be to generate palm that are grown faster, that are resistant to threats, and from which we can take the knowledge that we would learn from the date palm and transfer it into crops. And of course, ideally, what we would like to do is to establish a date palm breeding program here at KAUST. And of course, this is in collaboration with many entities in the kingdom, and one of the our most important collaborator is the Date Palm Center of Excellence in Al-Hasa, which belongs to the Ministry of Miwa. And what we would like to achieve at the end is to have a palm that can grow by combining all the approaches that I have just told you now, is to have palms that grow faster, make much fruit earlier, and that can be used for food security, because we like it to not. Many people are becoming either vegetarians or vegan. So, and with the nutritional values of date palm, so the dates can be a good substitute for other food. So what is the impact for the kingdom? So first, building tools. We are now in 21st century, and there is not a single tool that we could use to do research on date palm. So I think it's about time that we can start building these resources, and time to get to work, basically. So how to do that? We want to generate molecular and biotechnological technological tools, and again, back to basic science. We have to start from the beginning. Date palm have been neglected, and the date palm research has been neglected for so many years. We only, so far, there are a lot of genomics, which is very, very informative. There is a lot of omics, there is a lot of physiological, but there is not really hardcore basic research that can be translated into meaningful outcomes. So I think it's about time that we wake up and start working hard and doing what we need to do if we want the date palm agriculture to continue and to move faster, and if we want the date to be a substitute for food security. Then, of course, what we want to do is to preserve local cultivars. Many cultivars have been lost to our generation, and by using this fast feed program, I think we can achieve this goal. And of course, transfer the knowledge to other crops. How do we do that? So we have date that can grow under saline conditions. If we know what kind of traits are attributing these growth into these during these conditions, then we can immediately translate them into tomato, potato, salad, you name it. And that's what I wanted to share with you this morning. So thank you very much for listening, and welcome to KAUST.
Okay, thank you to all of those 4 speakers. We now have an open question and answering session. Perhaps if you've got some questions for any of the speakers, you could put your hand up and we can get the microphone to you. But while you're thinking about some questions, perhaps I could start. So you have a microphone in front of you. You just press the button on the side once, the light will turn green, and then you can speak into the microphone. So one of my questions is, once you've developed up new crop varieties, how do you integrate those into existing farm practices? How do you get those new varieties out into existing farm practices? Simone, maybe.
It's a good question, Paul. And I think, again, I showed it in my slides. We are talking about developing new cropping systems. There's different ways. For example, this semi-domesticated fonio millet I mentioned, that's already consumed. You can just grow it and use it. Actually, we've already been contacted by the Royal Commission for Al-Ula development because they're interested in this crop to test it. Of course, when we switch to saltwater-based agriculture, that will require some innovation, how we grow these crops. But again, I mean, one of the best places to do innovation is Saudi Arabia, right? I'm not worried about, developing new machinery, for example, and how you plant or how you harvest these new crops. I think that the major thing here is really get the new crops ready. Okay.
And perhaps a follow-up question while we're still waiting for other questions from the floor. Oh, we'll come to you. I'll have one more question and then we'll come to this questioner. But if they are a radically different crop species, culturally, how do you integrate those new crop species into someone's diet?
Yeah, okay, that's another good question. I'm looking at my student Hanin over there because Hanin has already used fonio millet and cooked traditional recipes, Saudi recipes, including date cakes. So talk to her, it worked beautifully. And again, we talk about this wild rice that Rod Wing is working on. I mean, again, at the end of the day, it will probably just be a salt water-based rice. And if you cook it and taste it, it probably doesn't taste too different from the existing rice. So I think that's the direction we want to go, so that at the end of the day, the consumer doesn't get something that is totally different.
Thank you very much. We have a question here, sir. Can you turn this on, please? Yes, thank you.
Yes, I think we heard very great talks from the panelists. It's really, they are really exciting subjects. My question is related to the continuity of this kind of research. This is very important because we have seen in the past some great research efforts, but only lasted for a short period and then disappeared. I think this is maybe a question as important as the research itself. As scientists sometimes focus too much on research to the point where they forget what's after research. You know, how to make this research continuous for a long time to have the impact. This is the first part. The second part is how to quickly implement this kind of research, how to apply it
in order to improve the legitimacy of what the researchers are doing. This is very important because you can get a lot of support, as we know that that research has great impact, but not enough of it is being done. So my question is how to make this kind of research continuous for a long time to have the largest impact, and how to quickly implement it to improve the legitimacy of the society continue supporting this kind of research.
Good question. Thank you. Professor Salim.
Thank you very much for this very important question. I mean, we have been speaking about completely different topics, so there will be different strategies actually to move on and to get the impact that we want to achieve. I believe a partnership with private sector is very important. That is actually the motor that would drive such research and make their fruits a reality. We have been now partnering with UPL, that is the third player, I think the globally third player in agrochemistry. We are working now on formulation, we are working on application and so on. In principle, if you are working at a university, you need actually a private sector partner that shows you also how the real world looks like.
I just want to add something else is education is important. So if we want somebody, we need somebody to carry on the torch. And I think we have great examples. We have Nahid, we have Hanin, we have Fatma. So I think whatever we produce now, it's going to be, let's say, inherited by the young generation, and these are the ones who are going to carry on the research. So it's not going to be a 2, 3, 4 years research, but decades of research. And it's all up of you guys, the Saudi young generation, to carry on the torch.
Thank you. We have another question over here.
Hi. First of all, I'm not that fluent in English, so if I mistake, allow me to mistake, please. And my question to Dr. Ikram, that if we will accelerating the growth of date palm, it can be, is there any chance to lose its vitamins or values? And another question is why Ajwa is there's a good growth in Al Medina and Oman nut. If we do a genetic changes in the date palm, is there any effect on the environment of the land can cause?
So these are really good questions. Thanks for asking them. Of course, we don't know. So what we try to do is to mimic the environment by recreating all the compounds that are present in soil. For example, we're adding microbiome, we're using growth regulators. So I think our first goal is to make them grow faster. And then using basic features like how date palm are flowering, how to make them flower earlier, how to improve fruits by looking at the fruit genes. So with these tools, we can make sure that we are ensuring the quality of what we are going to produce. Then, what was the other question?
If we grow the Ajwa palm in another way that is not adapted, it can be affect the environment, the land?
No.
So
no.
can I just repeat the question?
Yes.
Yeah. So you want to?
Yeah. So the question is, if we grow, for example, plant like Ajwa in another place, which is like in Oman, is it going to be harmful to the environment? Of course not. I mean, people have been transferring plants from one land to another for generation. An example is that we have medjool that have been transferred from Africa to California. There is also a large plantation of dates in Elche, the south of Spain, and there is no harm, and that has been for centuries already. So I think there is no impact on that.
Thank you. I think we have a question over here.
Hello. Thank you very much. Again, about the date palm issue. Don't you think there's also like other important factors that need to be addressed in the kingdom, along with the plant genetics to boost the plant productivity, such as like a soil fertilizer, technological use, and so on, so the kingdom actually can grow its production?
But we prefer not to apply too much fertilizer, because looking at the long-term goal, if you put too much fertilizer, it's going to impact negatively the soil, of course. So ideally, by just reconstituting the native soil, or by adding things that are already in soil. For instance, the good example is the microbiome project from Professor Heribert's here. So by just understanding which bacteria is able to improve tolerance to salinity or to drought, you can solve this problem without the use of fertilizer.
Sir.
Thank you. Dr. Nahid, I think the juniper dieback problem is, I remember it was more than 16 years ago, or almost that. So I think it's very severe in some area. So I think solving this problem, it looks to me a priority at this stage. Also, a second question for Dr. Ikram. Do you think one day we will have a dwarf date palm tree?
I don't see why not. You can just manipulate the genes that are accelerating the cell elongation of the plant. It's possible, but
I think
with a lot of patience.
I think it will have a great impact in the economy of date palm production in Saudi Arabia. Thank you.
Question.
Still, Nahid.
Yeah, so your question is about the juniper dieback. I know that I think in 2006 there was a rehabilitation program by the Ministry of Agriculture to these juniper trees, but unfortunately that was like a jump to a forward stage. The first thing that we need to do is diagnosis. So first we need to know why these junipers are dying back. Is it like a disease from a fungal effect, or is it the climate change? Is it the drought, or is it other effects like biotic or abiotic? So the most important thing is the diagnosis. And what we are doing now would help a lot to understand why these junipers are dying back, simply by doing population genetics to study like different juniper populations, the healthy ones, and compare it to the ones that are dying back. And if we see the genomic variation, for example, that would also help us address the problem. But again, the first thing is diagnosis. I know that the rehabilitation program that was done by the Ministry of Agriculture, it was just there without understanding why these trees are dying back. So that's the first goal, is trying to understand why, and then apply the appropriate solution to that.
Thank you. Jonathan?
Thanks very much. Yeah, thanks.
A bit closer, please.
Thanks to all the panelists. So plant breeding is classically not just about genetics, but genotype environment interaction. I guess this question is mostly for Simon, be interested in anybody else's comments as well. So are you trying to envision not just your ideal genotype, but where to put it and what kind of way of managing water would be sustainable? We were hearing from Matt McCabe and others yesterday about how unsustainable it is to be just mining from aquifers. So, if you grew, for example, that very drought heat tolerant millet somewhere around here, could you conceivably actually sustainably or at least at an adequate, at a tolerable cost, use desal to irrigate it by drip irrigation, just sporadically? So are you trying to envisage not just the genotypes you want to produce, but also where you want to put it and how you might manage that environment that you're going to put it in?
Yeah, of course, excellent comments on it. And I think, yes, of course. Of course, the main focus at the moment is on the genotypes, on the plants. Again, we also go out and try to identify new wild crop relatives that we could incorporate into this program. But yes, in the long term, of course, we need to think of how we, again, come up with new cropping systems. Is diesel going to be economically viable if we install drip irrigation? Again, for example, Fonio millet, I see there's something that is sort of, it could be deployed relatively quickly because it's already grown for food, and it can reduce the freshwater footprint. It will not, you will still need fresh water. And then again, I think we have other crop species like this wild rice or saliconia that are probably a bit more long term, but they could really significantly, maybe even to a large extent, get rid of all the fresh water in the agricultural system. Of course, we need to think how we deploy, what's the best way to deploy it. I can't give you the answer if diesel is going to be economically viable for this type of agriculture. We would have to look at it. But again, I mean, we're in a place, we have McCabe's research, we have a water desalination center, so we can just collaborate and see what's the best way to implement all of this.
Thank you. Last question here, sir.
Yes, thank you very much for this wonderful talks. I actually have two questions. One is for Dr. Salim, and it's a quick one. Dr. Salim, do you believe that those strigolactones can work with other invading plants, such as the prosopis and the Phragmites australis? It's spreading enormously in the kingdom in special locations. So do you believe that there is a solution like suicidal germination or other solution for such cases? This is my first question. My second question is for Dr.
Simon,
I believe. Dr. Simon, thank you very much. And I would like to just hear about how the consumer acceptance for those imported new crops. How would the people would change their habits instead of eating rice, mandi, madbi, whatever, to new food? How do you think this is going to impact the economy and impact the socio-economy on the other hand? Thank you.
Professor Salim first.
Thank you very much for this excellent question. Strigolactone themselves would not be very useful. However, we have a different chemistry. We have now a different principle. When we apply this chemistry, then we push the germination of all seeds. So if you want to clean off the soil of seeds of weeds, you could in principle do that before you plant your host, or before you plant your crop. We didn't publish this yet, but that is going to be also a breakthrough in our project. Because major issue when you work with weeds is that is a wild population. There is no synchronization in anything. You have a huge diversity in time of germination, ability to germinate, growth, phenotype, and so on. And we have something that actually push the germination of all seeds. Thank you very much.
Thank you, Professor Simon.
I mean, that was a massive question. Thank you. So, I mean, again, I think, we talked a lot about it. How long will the freshwater reservoirs of Saudi last? And we talk, maybe decades to maybe a century, right? So I think we really need a disruptive long-term solution. And this is what this project is about, right? It's disruptive. It will come up with something new. And I see it as an opportunity. Again, how will consumers react to it? One way really is that, again, if it's a salt-tolerant rice that can grow in salt water, it's still a rice. And of course, at the end of the day, the aim is that you can breed it and have its quality to a level that is comparable to the rice that we grow today. So maybe there's not even that much change for consumer. Again, Fonio millet, we can already use it. We used it in traditional Saudi recipes, and you can substitute it, for example, for couscous. It's quite similar. And of course, there might also be new types of crops that will have a different taste and that will require that consumers change their behavior. But I think, again, this is necessary given the challenges we have. And I think it's a huge opportunity also to develop an entire new, not just the cropping systems, but also the downstream processing and maybe marketing.
Thank you, everybody.
Just one quick things because we have heard a lot about consumer perception. I think the culture is changing. 50 years ago, if you come to Saudi Arabia and you say, let's go and eat sushi, nobody will go and eat it. But now I think we're getting more open toward accepting new varieties, new crops, new food in the sector. So I think it's not going to be a problem.
Thank you very much.
I just want to add something. At the end, it's a question of marketing.
Yeah.
And fonio and millets in general are very nutritious. And if you highlight that, if you tell the consumer how nutritious and how useful and beneficial these new crops are, then he will actually take them.
Thank you, Professor Salim. We are very soon about to have a very nutritious lunch, but before we do so, can you please thank all of our wonderful speakers for the session? Thank you. So I think what we heard from that session is that science can develop the solutions to pests and diseases and transform the productivity and resilience of food production in Saudi Arabia. So it has a really important role to play. But it is the ecosystem of science and translation and policy and commercialization that you need to ensure that the benefits of all that research and science is realized in the broader community. So fantastic set of presentations and some really strong messages for us to take away with. I need to explain to you what will happen after lunch because it's a little bit complex. So the session immediately after lunch will start at 1 o'clock. We'll go for 1 hour. There will be 4 different sessions. So you can choose which session you want to go through. There are 4 different rooms. The 4 sessions are food systems and education in Saudi Arabia. The edible schoolyard program will be in Hall 3A, which is just next door. The second program is saline agriculture for sustainable food security in Saudi Arabia. That's in Hall B, also next door, just here. The third session is controlled environmental agriculture for food security in Saudi Arabia. That will be here in this hall. And the fourth is regulatory frameworks to facilitate agricultural biotechnology. That will be in building 20 on level 2A. So you'll have to walk down some stairs, go across the bridge and then walk down some stairs to get to the balcony. There will be some people out there to help you if you get lost. You can also see the locations of those 4 programs on your program, on the online program. So if you can't find someone to talk to, you can go online and find out where they are, or you can just wander around and go into one randomly. At the completion of the focus sessions at 2 o'clock, we will have an afternoon tea break. The final period of the day will either be breakout sessions or it will be a tour of KAUST. The breakout sessions are by invitation only, so if you don't have an invitation, your opportunity is to go on the tour of KAUST. The breakout sessions are for the leading experts in various topics to come together and agree on the preferred solutions to the challenges proposed yesterday and bringing forward some of the work that's been done today. If you participate in these breakout sessions, you will know already the tour of the spin-outs. There will be some people here to guide people to buses, presumably. Okay, so people here to guide you to buses. We will reconvene in the main hall at 3:45 to hear a summary of the breakout sessions, if you're in those breakout sessions. And the summary will really set up the final discussion in the summit, which will happen tomorrow, which will explore how do we deliver. So the questions we were getting at the end of that session was, great, good science, how do we deliver this? So that's what day 3 is about. There will then be a reception in the library from 5 o'clock, and that will include a poster session. And that will conclude the proceedings for today. We will reconvene then tomorrow at 10 a.m. Right, so you all get an extra hour to sleep in or read emails. So it's 10 a.m., not 9 a.m. tomorrow. Please be registered and seated by 9:55. That's the final day of the summit where all the strands over the last 2 days will be brought together. And we will end up, we hope, with some common agreement at the end as to what the future strategy should be for building sustainable food security in the kingdom. Okay, so that was a lot of information. If you have any questions, ask the people with the green CDA t-shirts. They can help you. You can ask me or you can ask Rebecca. But now we will break for a lunch break. The next sessions will be those focus sessions in those 4 rooms that I described. And so please make your way to those rooms to start at 1 o'clock.